The next speaker is Nicolas Plachta. I met him 2 years ago at Northwestern, and I was fascinated with his research. He brings to life what used to have to see just happening in the uterus. He shows how is the development of the embryo in live imaging. You see how they grow up in front of you. He has been a world trotter. He is originally from Argentina, and then he moved to Switzerland, then he was in Australia, Singapore, and now is a professor at UPenn. So please welcome Nicolas.
Thank you so much, Miguel. Thank you, Joel, and everyone for this wonderful summit and the invitations. Fun. Great to be here, and let's see. Yep. So we're an imaging lab, and we use imaging to study embryonic development, especially the super early stages of mammalian development. We've been using the mouse embryo because it's similar to human, and it's just like a super common, useful, excellent laboratory model. But it's also a fundamentally wonderful system to do basic cell biology in vivo, in an intact physiological system where cells, which are initially round and completely undifferentiated, start to divide and start to make these decisions about which type of differentiation pathway they're going to follow. We can watch how they start to form the first tissue architecture or structures of tissue architecture, how they start to get into different positions in the embryo. So basically we use imaging to understand how an embryo forms, but also to understand how cells function in vivo in an intact developing environment. And we can get these embryos out of the uterus and culture them on top of microscopes. We use laser scanning microscopes, so we can culture them from the one cell when they just got fertilized until the blastocyst stage, which is just before implantation. That's the embryo forming the blastocyst at the end. And also because we work a lot with mouse embryos, we can genetically manipulate them. We can inject them with RNA, DNA, and all these things to make all structures inside the cells, proteins, subcellular compartments like the cell nucleus, the membranes, the cytoskeleton, fluorescently labeled. And we can do these time-lapse imaging studies where we image in 3 dimensions the whole embryo over time. And this is just a typical movie where we can see not only the cell-cell interactions and overall cell dynamics, but also we can see inside single cells at the more dynamic subcellular changes in the cytoskeleton, the cell nucleus, and so on. And that has been a powerful approach in the lab to discover especially random things we didn't know or expect that control embryonic development. And that's what I want to emphasize today. I want to show you a number of mechanisms and structures and even organelle kind of things that we run into or discover randomly using imaging in the mouse embryo. And then I want to show you how we start to move some of these imaging of embryos from the mouse to the human also. So we started working on those kind of like 3 major questions: how cells are deciding their lineage fate or identity, how they choose how they start to change shape and polarize, and how they get into different positions in the embryo. And the question about cell fate or lineage identity was very difficult because in other species, not mammals, for example, that's a worm embryo on the right, we know that even at the one-cell stage of development, there is some molecular asymmetries or molecular determinants which are asymmetrically localized and inherited during the first cell divisions during development that predict lineage fate. But in mouse or human embryos, that's a mouse embryo, until the 8-cell stage approximately, all the cells look very similar to each other, and it was very difficult to know which one will later produce more cells that end up contributing to the embryo proper or to the future trophectoderm, the outer layer that forms the placenta. And I skip all these details here, but we started to look at the dynamics and function of transcription factors, the key proteins that regulate gene expression in the embryo, and at their dynamics of DNA binding in the intact embryo. And we found that lineage patterning can be predicted super early, even at the 4-cell stage of development. So when you look at an embryo made of only 4 cells, we can already detect differences between the cells in the way that some transcription factors are binding on and off to DNA that predict the fate of these cells later on, if they're going to contribute more progeny to the inner part of the embryo, the pluripotent inner cell mass that makes the future fetus, or to the outer trophectoderm layer. So we started to gain some information about these early lineage decisions, but we wanted to understand how those processes are integrated with patterning and morphogenesis of the embryo, changes in cell shapes, changes in cell positions. So this is the first time cells start to change shape dramatically during development, which is during the time of embryo compaction. It happens around the 8-cell stage in the mouse embryo. And what happens here is that the cells they're initially very round, and they start to kind of squeeze and push against each other and form a compacted embryo morphology. And compaction is important for implantation later. And we didn't know what mechanisms exactly drive this compaction process. We knew that it has to depend on the action of cell adhesion molecules, the molecules that allows us to adhere and compact against each other. And the most important one in the early embryo is the cell adhesion molecule that's most important is E-cadherin or cadherins. Again, these are cell membrane receptors. So we started to image cadherins in the embryo. Again, these are live embryos. We took cadherins fused to GFP, the green fluorescent protein, so they're all green. And we found that the cells in the embryo are not just boring round structures. They actually have these long protrusions. These are like long cellular protrusions. We call them philopodia. And they extend from some cells in the embryo on top of the neighboring cells, and they don't sit there in just a static manner. When we do movies of these embryos, you can see they are dynamically extended and retracted, and we could show that these structures are used like kind of long arms that appear during the process of compaction that cells use to kind of bring each other closer, hug each other, and give the embryo its compacted morphology. So this was, again, a random cellular structure that we didn't imagine, that all of a sudden when you do live imaging appears in front of the microscope. And we think that's important not only for compaction, but also to understand cell biology in general, because almost every cell in our body uses cellular protrusions of different sizes and thickness for important things, like cells migrate in the body, connect with each other, establish neuronal synapses using all sorts of protrusions. And we don't have useful model systems to study how on earth cells grow these protrusions and how they regulate them in a physiological environment. So now we can do that in the intact embryo and look at how these things are regulated, also if they're used for transporting molecules and signals between cells. So that started to tell us a little bit about how cells start to change their shape for the first time and polarize. And the next question was how they start to change position. Because initially, until the 8-cell stage, more or less, all the cells are symmetrically arranged in the embryo. There is no inner mass. But by the 16-cell stage, some cells internalize, those ones in orange, and those 3 inner cells that internalize during that stage are actually going to form most of the inner cell mass, and we come mostly from those 3 inner cells. The cells that stay in the outer part will form the trophectoderm layer that will form the placenta. So that's the first very fundamental lineage differentiation during development. Are you going in, or are you staying out in the outer part of the embryo? And there was a textbook model that always suggested that some cells may be just dividing in a very spatially asymmetric manner. So as a direct result of this division, one daughter cell, it's kind of pushed inside the embryo directly. And we wanted to test that model. We wanted to visualize divisions in real time. So we worked with embryos where we use embryos that have a fluorescent marker in the membrane, so we can see all the cell contours. And then we came up with technologies to computationally segment every cell in the embryo, reconstruct in 3D every cell in the embryo. So this is a movie of of an embryo, it's live, a cell is dividing, we segmented them, and you see this cell in yellow, it's dividing, but it's not undergoing that textbook model division, super asymmetric. It's dividing actually more along the surface of the embryo, and then only one of the daughter cells, the one in red, starts to gradually internalize. And the way that cell is actually internalizing is the parental cell divides along the surface, and then one of them internalizes gradually over time. So the formation of the inner cell mass, it's not driven by these very asymmetric divisions. It's mostly driven by this two-step process where cells divide along the surface and then gradually internalize. And we more recently started to work out why some cells internalize and others don't in the embryo. I skip all the details, but we found that this is determined by the organization of the mitotic spindle, the main microtubule apparatus that drives the segregation of the chromosomes during mitosis, but also it's a structure that can change the orientation and asymmetric inheritance of things during cell division. So we started to understand a little bit about morphogenesis and changes in cell shape and position, looking at mostly mechanical forces and the cytoskeleton, and we wanted to dig deeper into the cytoskeleton in the embryo. And we looked a little more first at the actin cytoskeleton that forms mostly the cell cortex. And what we found is that, again, random discoveries, just around the 16-cell stage, some cells in the embryo, most of them, form these really beautiful actin structures at the cortex, at the apical part. So these are very beautiful ring-like structures that form in the outer cortex that faces the outside, the zona pellucida. And when we see structures like this, and people have seen them before, but when we look at them, we think, they look at actin cables. These things must be used for like squeezing or shrinking things, like the cytokinetic furrow that forms during cell division establishes a ring like this. They should have myosins on them, but when we look at them using live imaging, we found that they don't have any myosins on them, and they're actually not closing or constricting. They do actually the opposite. These are expanding rings. You see they're growing over the entire surface of the embryo until they come into contact with each other, and you can see here a few snapshots of those rings, which are small, and they grow and grow and grow over the entire surface, and they start to kind of like fuse or zip with each other along the cell-cell junctions where the cells touch each other. And that was exciting because this happens just before the mouse and the human embryo form the blastocyst cavity. And to form that cavity, the cells need to establish the first permeability barrier so they can pump fluid inside and expand that pressurized cavity. And that's how the blastocyst forms, by formation of this seal permeability barrier. And we think that this zippering of actin rings is providing the mechanism to seal that embryo. You can see here, these are two actin rings that just came into contact with each other between two cells, and we can literally see the zippering process in real time. And in green, we're following a key protein, it's called zona occludens, that forms the tight junctions, which are kind of like the zipper, the teeth of a zipper. So we can literally see how this zippering process is sealing the embryo, and if we can disrupt this genetically or with other ways, these embryos do not become sealed. We don't form a blastocyst cavity. So again, it's a random discovery of these structures we didn't expect. They expand, and this zippering seems to be driving the sealing of the embryo for blastocyst formation. So we made some progress looking at actin, and we wanted to focus in the other two major components of the cytoskeleton. Again, the cytoskeleton is made of different filamentous networks. Actin is one of the most famous ones. Then we have microtubules and intermediate filaments. So microtubules are also beautiful, and it was a problem because in many cells, we have an organelle which is called the centrosome, which acts as the main microtubule organizing center. And most of the microtubule cytoskeleton is organized by this centrosome. But mouse embryos, they don't have centrosomes. They only establish them after implantation, apparently. And people always thought that microtubules may be very randomly organized in the embryo. So again, we were imaging microtubules in the lab, and we found randomly this thing there that shows that microtubules are not completely randomly organized. There is something that is bundling microtubules there. Now, that thing there is the bridge that forms between cells during division, the cytokinetic bridge. And what happens in most cells in our body, in literally every cell, when they divide, they form the mitotic spindle, then it forms this bridge, and at the end, that bridge needs to be cut. It's called abscission, and that's how we end up with two happy independent cells. But when we image microtubules in the embryo, now the microtubules are in orange, that doesn't happen. In the mouse and also in the human embryo, cells divide, and they don't abscise, they don't cut the bridge, they stay connected with each other. And we can look at any cell and any cell division in the embryo, and we always see sister cells connected by this microtubule bridge. So actually in the embryo, every time that we think we have like a fully independent cell, it's still mechanically connected via this bridge to the sister cell. In fact, every time we look at those bridges, we know which cell is sister with which in the embryo. And here we go and we do imaging of the growth of microtubules at those bridges. So this is almost a real-time movie that shows us the growing tips of microtubules. And what this shows us is that those bridges are working like centrosomes. They are promoting the growth of microtubules inside the cell. So the embryo uses these unusual ways of microtubule organization, which is different from later on in the body, and also the cells stay connected. So we learn quite a lot about how different cellular organization can be in the embryo compared to all other, let's say, more normal cells in the body. And I just want to finish with intermediate filaments, because this is the other third major component of the cytoskeleton that we knew a lot less about. There are many classes of intermediate filaments, but in embryos, there is only one class of intermediate filaments in the cytoplasm, which are keratins. So keratins are famous in the skin, the hair, but they're also present, and they're a famous marker of the trophectoderm, the outer epithelial layer of the embryo. And here you have mouse and human embryos, and they're a beautiful marker of the trophectoderm. You see how they form this really nice network in all the trophectoderm cells. But when we look at embryos earlier during development, we see that you have mouse embryos on top and human embryos at the bottom. Some cells start to assemble this keratin network before others during development. And when we see this heterogeneities, we're interested what's different between those cells. And not only that, but when cells divide during these mid stages, the cells that will remain in the outer layer and be part of the trophectoderm actually inherit all the keratins, and the cell that will later internalize to form the inner mass doesn't inherit much of these keratins. So this is probably one of the first examples of large cellular component that is asymmetrically inherited during division, and it predicts which cell or the fate of the cells, which one will be in, which one will be out in the embryo. And I skip all these details, but keratins are not only predictors of fate, but they're also functionally important because they help to stabilize the polarity of the outer cell and to regulate some of the key transcription factors that specify or activate the trophectoderm genes, like especially CDX2 and many downstream genes. So it's a clear example of something that is asymmetrically inherited and predicts, but also starts to determine fate in the embryo. And it also shows us that when we look at trophectoderm cells, they're not all equal. Some start to adopt a trophectoderm identity before others during development. So I stopped this part here. This was just an overview where I wanted to emphasize how we can use imaging to discover, especially in a random way, many new mechanisms or structures that control how cells are making those differentiation decisions, how they're getting into different positions, how they start to polarize to form the early mammalian embryo. And I just want to finish with trying to show you how we could start to move some of this research from mouse to human embryos, because we have a big fertility problem, and one of the key challenges in the clinic, in the IVF clinic, is to decide which one of these human embryos should or shouldn't be transferred back, which one is developing more normally. And all the imaging that we can do in human embryos for the moment is using this normal microscopy or bright field approaches, which is really nice, but at one point, you don't really, this is a 2D plane, you don't really see how many cells are present in the embryo. Initially you do, but as they start to compact and all of that, it's hard to see how many cells are there, their morphology, etc. And it would be great if we could image the embryo in 3 dimensions with fluorescence like we've been doing in the mouse. It's a problem to do that because all the mouse work we've been doing is using genetic manipulations, where we make the embryos express all these fluorescent proteins, which is difficult to do in human. In many places, you cannot even do a genetic manipulation in a human embryo. And also, many of the embryos donated for research come too late at the morula or blastocyst stage, where you cannot even microinject a large cell in the embryo. So we started to use different fluorescent dyes that companies have been, and chemists have been developing, that label different structures in cells fluorescently, and some of them are compatible with live imaging, and some of them started to work well, especially one that labels actin, so we can see the entire contour of the cells, and another one that labels DNA, so we can see the nucleus of every cell in the embryo. So those are mouse embryos, and they have not been genetically manipulated. We didn't microinject them with anything. We just cultured them in the presence of these fluorescent dyes that label the DNA and the actin, and we can image them with fairly good resolution, close to what we get with genetic manipulations. And if we use them not too concentrated, they're fine, they're not toxic, we can make babies out of these embryos, and now we can start watching cell dynamics again in mouse and in human embryos without any genetic manipulations. We can follow much better the morphology of every cell, we can watch them dividing in real time. So for us, it's kind of like a special moment because it's the first time we can go into a human embryo live in 3 dimensions and image and resolve much better the morphology of every cell. We can see the inner mass there when we do this in silico computational cuts, and so we can infer much better the development, but also study the development of these embryos. So we got some press attention, which was very nice, because these are nice images of human embryos we're getting, but also we can now, because we can fluorescently label cells, we can quantitatively track key morphogenetic processes, like, for example, the dynamics of embryo compaction, and we started to see some interesting differences with the mouse embryos. We could start watching the division of cells in the embryo that will produce later inner and outer progeny. So we can start seeing how cells segregate into the inner cell mass and trophectoderm in the human embryo. And we can take this all the way to the time of hatching, when these embryos need to escape the zona pellucida here, that membrane that encapsulates them, and are ready to implant in the uterus. And I just want to show you with other things we could image and we could discover in human embryos, which are important in the clinic. So we started to look at aneuploidies in the embryo. So there are many diseases that come from aneuploidies. That happens when you lose some chromosomes, or you have extra chromosomes. Yeah, Down syndrome is very famous, and most of them come from these, from mitotic errors. This is a movie done in an intact embryo. You're looking at the chromatin or chromosomes during mitosis, and one of these chromosomes lags behind, and you will see ends up just floating there, and it's going to create a problem. Aneuploidies mostly come from this, from mitotic errors, when we have lagging chromosomes, for example, that do not get properly segregated, and they will form a micronucleus, and that's a big problem, because that cell may then have, it's going to be missing or having an extra chromosome. And now it's nice because we can watch and quantify and show these divisions and mitotic errors in real time. And indeed, there's quite a bit of them in the human and in the mouse embryo, more than we have in normal somatic tissues, which is interesting to know why errors are more common in early development. But what we also found that, again, we didn't expect, this is not a cell undergoing division. This is the nucleus of a cell in interphase. It's not dividing, it's not in mitosis. And some cells in the embryo start to release or lose some DNA directly into the cytoplasm. It's not an artifact. We can see this with many different labels against DNA and chromatin and other nuclear markers. What happens is that just a few of cells in the trophectoderm at the blastocyst stage, like 2 to 4%, release or shed some DNA directly into the cytoplasm. Again, this is not a cell that is dividing or in mitosis. You can see here in real time a cell, it divides, and you can see there are lagging, it's before division. It's shedding some DNA into the cytoplasm, and then you can see that structure gets inherited by one of the daughter cells. So this is not coming from a mitotic error. This is happening during interphase. And we think that the mechanism, it has a mechanical explanation because it happens at the blastocyst stage, mostly in the trophectoderm. And what happens at this stage is that initially the cells in the embryo are very comfortable, the nucleus is very round, but when the blastocyst cavity expands, it generates a lot of pressure that kind of sandwiches or squeezes the trophectoderm cells and the nucleus of those cells. It becomes really squeezed. And what we think is that some trophectoderm cells have a more fragile nucleus that can undergo this transient rupture and release some DNA, literally lose some DNA into the cytoplasm. It's not a very strange, weird thing that we only see in the embryo. There are some beautiful studies in culture cells where they make them migrate through very narrow nanogaps, and the nucleus also transiently breaks, and these cells, the nucleus loses some DNA and nuclear material, and then it quickly seals back. So we think the same mechanical stress is causing that in the blastocyst, in a few cells during normal development, but also that may be important in the clinic when we do biopsies of the trophectoderm to test for aneuploidies, because here we're mechanically sucking the trophectoderm, and you can see we are deforming a lot those cells, and that generates a lot of mechanical stress. So we did biopsies in the lab, and I don't go into a lot of details, but basically we also see that the biopsy itself can cause some nuclei in the trophectoderm to deform and to lose some DNA. And so basically, it could be that some of the aneuploidies we detect and the mosaicism we detect comes not only from mitotic errors and aneuploidies, but also from mechanically stressing the cells and causing them to lose some DNA into the cytoplasm. So I stop here. Super, super wonderful group of people, phenomenal group. They do all the work in the lab, and all the human embryo work we do in collaboration with the groups of Denis Sakas in Boston and Carlos Simon in Spain. Thank you so much.